Hi there, students. This is another version of the online videos I've doing before, but they are created solely by me, the original creations, and I wanted to go over the topic of the HFR strain. And essentially it has the fDNA is this green blob here, and it's integrated into the bacterial genome, right? And what's special is where the fDNA gets integrated to in the genome, different outcomes can occur, for example, and also the orientation of the fDNA matters as well. So let's look at the fDNA orientation. So when the orientation starts and goes to the right, then and then methionine would be the first DNA to go transfer. So you can see there methionine would be first. That's the arrow. But when the orientation is this way, then the lactose gene will go first there. So the different orientations of fDNA will also control what genes come out first in conjugation. So that's one thing to remember. And then different integration locations of the fDNA will result in different gene ordering and conjugation experiments too. So if this guy started transferring its DNA during the conjugation, then either serine would come out first, or this GAL will come out first too. So this was sort of a big mental leap a couple students got when they came to discussion, was that the orientations of the fDNA and integration locations really determine the gene ordering. So, and also it's absolutely necessary for recombination to occur to make bacteria with new properties. So writing out this example is you would have your donor that's Rh minus and Met plus, methionine plus. So it can't grow in the absence of arginine, but it can grow fine in the absence of methionine added. So it can make its own methionine, but this guy cannot make its own arginine. And then the recipient eventually gets this chunk of DNA, and it's methionine plus the green part, and that exchanges with the methionine minus part of this recipient. So it's these are two different equili, and it's not F plus, but it got some DNA. And then it recombined, so the DNA needs to go in and then recombine. And then after recombination occurred, then this is what it looks like. There's a Met minus is on this chunk, and Met plus is on that chunk. And then this is the genomic chromosome of the bacteria. The genomic circular DNA is here. I just drew it simple. And then you would plate the cells, and these guys are called, and then you plate all these cells on special agar plates called methionine minus Rh plus agar plates, and then the bacteria that grow called exconjugants. So another way to show this information is here. You make a little graph, a grid. You clearly identify the donor, the recipient, and then exconjugant. So is the donor F plus? Yes, it's F plus. Is the recipient F plus? No. Is the exconjugant F plus? No. And also exoconjugates are generally rare, but no, it's not F plus. And again, this is with the HFR strain where it's genomically located, like up here, not the strain where the fDNA is on the plasmid. And then you ask, can it grow with the added methionine? So without adding methionine. So the answer is yes. This guy has the Met gene and it's functional, so it could grow in the absence of methionine. But this guy is methionine minus over here, so it cannot grow in the absence of methionine. So that's the recipient, so the answer is no. And then you ask, does the donor, can it grow with arginine? Well, it's Rh minus, so it cannot grow in that without adding arginine. It has a dysfunctional arginine gene that does not allow it to make any arginine for the cell to grow, and arginine is pretty important, it's essential. So without arginine, the bacteria can't grow. So the donor cannot grow without added arginine, but the recipient can grow with arginine because it already has the arginine gene up here, it's positive. So that's the difference between the donor and the recipient. Arg minus methionine plus, methionine minus arg plus. And now if you now you ask, can the bacteria grow on an agar plate without arginine and methionine? And their answer would be no, and the recipient would be no, because it needs arginine, but this guy needs methionine. So then you get these special exoconjugants that are rare. That's like these guys, the small dots, and that they actually look like this. They're methionine plus arginine plus. So the exoconjugates can grow only on agar without arginine and methionine. Oops. So that's pretty cool. You know, you can see this graph here that it goes, you know, yes, no, no, yes, no, no, yes. So this is an example of selection. Remember, selection versus screening. So this is an example of selection. The exoconjugant can grow, yes. It can also grow on all these different media conditions, but you won't be able to distinguish it from either the donor or the recipient. So the only way to distinguish between the donor and the recipient and the exoconjugate, which are these guys right there, I guess right here too, is if you grow on media that agar plates generally that have no arginine and no methionine. So that's pretty cool, and that's an example of how the whole exoconjugate thing, conjugation happened. Yeah, I'm not sure. Yeah, I'm really actually still confused on what's going to be on the exam, because we went over a lot of material, you know, but in summary, the orientation of fDNA, orientation and when it starts to replicate and form single-stranded determines the gene order, either methionine or lactose in this case. And then different locations, different integration locations also impart the property of what the gene order is. So I think that's sort of interesting. And then it's absolutely necessary for recombination to occur to make the bacteria with the new properties. That's the non-parental progeny. So hopefully this clarified what was going on. And if you have trouble, just try keeping track of everything with these little grids and tables here on the lower left hand corner. And I gotta thank the professors for pointing out the fact that I need to probably copyright and just make a notice of this original material, you know, and that they were actually protecting me to not get in trouble. So I have to thank the professors for protecting me and not getting in trouble, and therefore I just made my original creation here with the pen. So these sort of problems are pretty easy, and hopefully this clarifies some of the mistakes, some of the misunderstandings, or adds to what the professor and the book has already added. So this is just another added option, and I thank you for your attention, and I'll see you next week.